Chapter 11, Topic 11.2, The Progress of an Infection. In this topic, we're going to be discussing whether a disease will result or not, given certain situations. We're going to discuss portals of entry, attaching, becoming established, causing disease, progress of infection and disease, signs and symptoms, portals of exit, persistence and pathological conditions, what happens in your body, reservoirs, acquisition and transmission, healthcare-associated infection, universal blood precautions, and Koch's postulates. The learning objectives for this topic are to be able to differentiate between pathogenicity and virulence, define opportunism, list the steps a microbe has to take to get to the point where it can cause disease, list several portals of entry, define infectious dose, describe 3 ways microbes cause tissue damage, differentiate between endotoxins and exotoxins, provide a definition of virulence factors, draw a diagram of the stages of disease in a human, list several different modes of transmission of infectious agents, define healthcare-associated infection, and list the 3 most common types, list Koch's postulates and when they might not be appropriate in establishing causation. You are exposed to bacteria and other microbes constantly from the day of your birth, but most of us get sick rarely. Why is that? Several factors need to come into play before disease will result. Let's go over some of the examples from the textbook. I'll be using specific examples to make it more concrete. In this first scenario, let's say the microbe is Bacillus subtilis, a common soil bacterium. So you're riding this bus and you breathe in B. subtilis in the dust in the air. B. subtilis has very low virulence. Really doesn't cause disease very often in humans. You don't breathe in all that many. If you breathed in a lot, your lungs would object to that much dust and you'd have a coughing fit, physically expelling the dust. B. subtilis is introduced to you through a portal of entry that it doesn't specifically like. It gets stuck in the mucus in the lungs and it's saying, "Whoa, soil is never this sticky." Mucus is a non-specific form of immunity, by the way, and it stops the B. subtilis from spreading. Also, you've breathed in dust every day of your life and you have antibodies, or specific immunity, to B. subtilis. The antibodies are in the mucus and finish off the bacteria. Besides all that, you are in good health. Being in good general health means that energy resources can be diverted to immunity, like making the right amount of mucus and making antibodies. The result is that B. subtilis is killed or passed through without you even noticing. Let's look at this next one. We're going to use rhinovirus as the example, which causes the common cold. Rhinovirus has a fairly high virulence, meaning that it's pretty good at its job of causing the common cold. So you are riding the bus and a kid sneezes in your face. You breathe in a moderately high number of rhinoviruses and it enters your sinuses. The sinuses are the preferred portal of entry, meaning when the rhinoviruses hit your sinuses, they say, "I have come home. Mucus, help me stick to these lovely cells." So from the point of view of the rhinovirus, everything is going well. But you happen to be genetically resistant to this type of rhinovirus. And you've had this rhinovirus in your sinuses before. So you have antibodies against it. You have a good level of health, so you've been making the proper amount of mucus with antibodies in it. So the antibodies cover the rhinoviruses so the viruses can't stick to your sinus cells and you don't get sick. Let's skip this third scenario and move on to the fourth one. With this last one, we're going to look at measles virus as the example. The virus that causes measles is one of the most virulent pathogens on the planet. It is very good at causing measles, and measles is a serious disease. It doesn't need very many virus particles to cause disease. So when that kid sneezes in your face on the bus, you only need to breathe in 5 viruses to be at risk. Measles virus is passed through the respiratory system, so the virus likes entering your sinuses. Everything is go from the virus's point of view. Now let's say you haven't had measles, nor have you been vaccinated, so you haven't been exposed to the measles virus and you don't have antibodies to it. And you are working full-time and you're going to school, so you haven't gotten the sleep that you need, so your health isn't at its peak. Your body hasn't been producing enough mucus to stop a big-time pathogen like the measles virus. The virus gets in, starts replicating in your cells. Soon, you pop out in spots and you have a fever. So, for disease to result, all of these factors interact to cause disease. It should be reassuring that for most of us, most of the time, we don't get sick. I've mentioned the portals of entry several times during the last slide. Portals of entry are ways the microbe can get into a human, like shown in this figure. All of these, though, break down into three basic portals: the mucous membranes, skin, and parenchyma. For mucous membranes, we are talking about the insides of your respiratory tract, intestinal tract, reproductive and urinary tracts, and so on. For most microbes, the skin has to be broken for microbes to get past, which is why we use Band-Aids to cover the break until you develop a scab, at least in lab anyway. For parenchymal routes, that is when the microbes are injected past the skin. That can be anything from a non-sterile surgical instrument to an insect bite. All pathogens have a preferred portal of entry. A pathogen that specializes in GI tract diseases isn't going to be happy in the respiratory tract. It wants to enter through the mouth. Once it's inside its preferred portal of entry, the pathogen has to be able to grab a hold. Bacteria, parasites, and viruses don't have hands and fingers. They grab a hold by binding adhesion molecules on the pathogen with receptor molecules on the target cell. Now, the target cell doesn't make the receptor for the pathogen to grab a hold of. The receptor is doing some other job for the cell, and the pathogen takes over for its own evil purposes. Once it is attached, the pathogen next needs to become established. To do this, the pathogen needs to evade the immune system while boosting its numbers. These first two refer back to Chapter 3. How do capsules help a pathogen become established? It prevents phagocytosis by white blood cells. If they aren't being eaten very fast, the pathogen can expand its numbers. What cell wall components am I talking about here? Acid-fast bacteria have a waxy substance, mycolic acid, which makes them hard to phagocytize as well. Now, this last one, antigenic variation, refers to how a pathogen can change its appearance to the immune system, so past antibodies don't work on it. There are other ways a pathogen can become established that are covered in the textbook, so make sure you understand how they help the pathogen. Now that the pathogen has become established, you generally start feeling the effects of this. You start feeling ill. There are three major ways that microorganisms damage their host. Directly, through the action of enzymes, directly through the action of toxins, and indirectly by inducing the host's defenses to respond excessively or inappropriately. Let's go over the enzymes first. Enzymes are released by bacteria and some protists to break down macromolecules in the environment until the macromolecules are small enough for those molecules to be transported through the cell envelope. Well, pathogens do the same thing. They break down your cells and tissues. This is to release nutrients to the pathogen, and also, in the case of this diagram, to help the pathogen spread within a tissue and an organ. Some pathogens release toxins. Some toxins affect the nervous system, such as the tetanospasm produced by Clostridium tetani. Some exotoxins cause vomiting, which helps the pathogen spread, and on and on. Some bacteria have molecules as a part of their cells which are toxic to the host when the cell dies, and the endotoxin, endo for in, is released. Remember LPS from Chapter 3? It is an endotoxin. Sometimes we are our own worst enemy. For example, when Streptococcus pneumoniae establishes within the lungs, if they have a capsule, white blood cells have a hard time phagocytizing them. So your system keeps sending signals that say, we need more help here. More and more white blood cells arrive through the leaky capillaries, which also allows more fluid to enter the tissue. This is part of inflammation, by the way, which we will be covering in later chapters. As fluid builds up in the lungs, we call this pneumonia. Sometimes these elements do double duty. For example, LPS is an endotoxin that can cause an excessive immune response. This table covers the differential characteristics of bacterial exotoxins and endotoxin. or ways to tell the toxins apart. I'll leave you to read over it on your own, but I will be asking questions about these characteristics. Here is a flow chart that summarizes the progress of infection and disease. It is a very good summary of what we've covered so far, so make sure that you understand it. When you get sick, you know it. Clinically, we differentiate between warning signs that we can measure, called signs, and those signals that only the patient experiences, called symptoms. Some examples of signs would be fever, vomiting, and rash. Think of a sign as being a signal of illness that is hard to fake to get out of going to school. A symptom, on the other hand, would be fairly easy to fake since only you feel it. So feeling hot, nausea, and itching would be examples of symptoms. Inflammation causes some classic signs and symptoms, namely heat, redness, swelling, and pain. In addition, granulomas and abscesses can result from long-term chronic infection. Lymphoadenitis, or swollen lymph nodes, is also a sign of inflammation. Signs of infection in the blood, or septicemia, can be an increase in the level of white blood cells, leukocytosis, or a decrease of the level of white blood cells, leukopenia. Another sign is culturing or detecting the pathogen in the blood. Septicemia is separated by what causes it. A septicemia caused by bacteria would be a bacteremia. Sometimes you are infected and do not have signs and symptoms. You go about your happy way without knowing you are being or have been infected. It's only when damage is caused or the immune system responds that we actually have disease. Well, let's finish the steps in the life of a pathogen. Once they've used you up, or if the immune system starts winning, the pathogen wants to leave and start over with a fresh host. They leave in ways that will put them in contact with their portal of entry. For example, respiratory viruses leave by the respiratory means. For some, the portal of entry is not only the best way to contact the portal of entry, but the natural consequence of anatomy. A GI tract pathogen goes in the mouth and goes out the anus. The GI tract is pretty much one way. Well, not if you're vomiting. Some pathogens leave colonies behind when they leave. Take, for example, chickenpox caused by varicella zoster virus. Once you've got it, you've always got it. It hangs out in your nerves, waiting for your immune system to drop off and start paying attention. Then it travels down the nerves and causes little blisters around where the nerve endings are. Speaking of traveling down the nerves, some patients with shingles or reactivated varicella zoster virus experience nerve damage. This causes pain for days or even months. This pain is known as a type of sequela, or long-term or permanent damage to tissues and organs. We can graph what's happening in the body. It goes from incubation through the period of illness, through a period of decline, into the period of convalescence or recovery. Does this look like a growth curve to you? It does to me. Of course, in illness, there's more going on than what's happening in the growth curve. For example, the period of decline isn't caused by running out of nutrients. Instead, it is caused by your immune system winning the battle against the pathogen, which causes a decline in pathogen numbers and a decline in the signs and symptoms. In order for an infectious agent to continue to exist and to be spread, it must have a permanent place to reside, or a reservoir. It's where the pathogen hangs out while it's waiting for your immune system to be on the other side of the body, so to speak. This picture shows some of the most common reservoirs of human pathogens, the most common one being other humans. Most human pathogens want to go from human to human. Make sure you go over the figure in the textbook about carrier states. Some can hang out in domesticated animals, though, like the sheep and the sheepdog. Some pathogens hang out in wild animals. I have a ground squirrel pictured here because plague is still found in the U.S. and it has a reservoir in ground squirrels. If you find a dead squirrel, don't go anywhere near it. The fleas that carry the plague pathogens can jump from the dead squirrel to you. I put the goose in because they are major reservoirs of influenza. Think bird flu. Not all reservoirs are alive. Water, wind, soil, dust can be reservoirs for some of the tougher pathogens. Some of the wimpier pathogens must go from fluid to fluid. If they can't go from human to human, they may use a biological vector. Think mosquitoes and the spread of West Nile virus in the United States. Imagine a mosquito buzzing around the head of the shepherd in the photo. For acquisition and transmission, I have some lovely curiosity clips. They are very well done, entertaining, and disgusting. Before I end here and leave you to watch those clips, I want to explain the ATP meter that the microbiologist uses. ATP doesn't last very long. Mainly it is found only in living cells. So measuring the amount of ATP is a good way to measure the numbers of living cells without actually having to culture them or count them.